Hello everyone. Today we will discuss photoperiodism. We will explore how different plants interpret and respond to changes in daylight, shedding light on the intricate mechanism behind this phenomena. So let's start. Photoperiodism is derived from the Greek word photos, that is light, and periods, that is the length of time. It refers to the plant's response to the timing of light and darkness. The most significant photoperiodic response observed in plants is flowering. However, other responses such as pigmentation, branching, tuberization, or dormancy can also occur. Then we have the nature of response, that photoperiodism encompasses the physiological changes that plants undergo in response to the relative length of day and night, known as the photoperiod. Now we will discuss about the discovery. The concept of photoperiodism emerged from the pioneering work of plant physiologists W. Garner and Allard in 1920. Conducted at the United States Department of Agriculture in Westville, Maryland, USA. Their research focused on investigating the growth and flowering patterns of two key plant species, that is Biloxi soybean or Glycine max, and the Maryland mammoth variety of tobacco or Nicotiana tabacum. Through their meticulous observation and experiments, Garner and Allard made a significant breakthrough by determining that the relative length of the day, known as the photoperiod, played a pivotal role in influencing plant growth and development. This discovery revolutionized our understanding of plant physiology and the environmental cues that regulate plant behavior. Furthermore, they noted the intriguing similarity between the responses of plants to photoperiods and circadian rhythm, both of which are intricately linked to cycles of light and darkness. This finding underscored the interconnectedness of these biological processes and provided valuable insights into the mechanism of governing plant responses to environmental stimuli. Overall, Garner and Allard groundbreaking research laid the foundation for the study of photoperiodism and its fundamental importance in plant biology. As you can see in the figure here, the Maryland mammoth mutant of tobacco on right compared with the wild type tobacco on the left side. Both plants were grown during summer in greenhouse. Numerous plant species exhibit flowering patterns correlated with the long days of summer. For many years, it was widely believed among plant physiologists that this correlation stemmed from the accumulation of photosynthetic products that is synthesized during these extended daylight periods. However, this hypothesis was rejected by the groundbreaking work of Whitman Garner and Henry Allard in the 1920 at the US Department of Agriculture Laboratories in Maryland. Garner and Allard research focused on a mutant variety of tobacco known as Maryland mammoth, which exhibited vigorous growth reaching heights of about 5 meters. It failed to flower under the prevailing summer conditions. Plants are categorized into three groups based on the duration of the photoperiod: the short day plant, long day plants, and day neutral plants. So, the primary difference between the long day and the short day plant lies in their flowering responses to varying day length. Long day plants initiate flowering only when the length of daylight exceeds a specific threshold known as the critical day length within a 24-hour cycle. In contrast, the short day plants require a day length that is shorter than the critical day length to induce flowering. This critical distinction in photoperiodic response mechanism governs the flowering behavior of plant and is crucial for understanding their adaptation to environmental conditions. Short day plants exhibits flowering response that are triggered either exclusively in short days known as qualitative short day plants or accelerated by shorter day length that is referred to as quantitative short day plants. These plants typically necessitate to relatively brief duration of daylight, usually around 8 to 10 hours, followed by an extended dark period spanning approximately 14 to 16 hours to initiate flowering. Often referred to as long night plants. Examples of short day plants include rice, coffee, soybean, tobacco, and chrysanthemum. The critical day length or photoperiod required for flowering varies among species. For instance, violet plants bloom when exposed to day length shorter than 11 hours, whereas chrysanthemum require day length shorter than 15 hours to induce flowering. Despite the species-specific differences in the critical day length, both violet plant and chrysanthemum are classified as short day plants because they fail to flower when exposed to day length exceeding their respective critical threshold of 11 or 15 hours. In short day plants, the uninterrupted dark period is crucial for flowering initiation. Disrupting this dark period with a brief exposure to red light of 660 to 665 nanometer wavelength prevents flowering in the short day plants. The most significant inhibition of flowering due to red light exposure typically occurs around the midpoint of the critical dark period. However, the suppressive impact of red light can be counteracted by the subsequent exposure to far red light, which is of about 730 to 735 nanometer wavelength. Interestingly, interruption the light period, or you can say the interrupting the light period with the red light does not hinder flowering in short day plant. Additionally, extending the continuous dark period can lead the early flowering initiation in these plants. As you can see here in the table, there are some examples of short day plants. It includes the winter rice, bryophyllum, chrysanthemum, cocklebur, cosmos, hemp, Japanese morning glory, calancho, lespedeza, morning glory, poinsettia, and violet. So now we will discuss about the long day plants. In short, it is called LDPs. They flower either exclusively in long days or have their flowering processes accelerated by longer daylight periods. These plants typically necessitate a prolonged period of daylight, typically lasting 14 to 16 hours within a 24-hour cycle to initiate flowering. They are also referred to as short night plants. In long day plants, the duration of the light exposure is crucial for flowering initiation. Flowering in these plants can be stimulated by either a brief exposure to red light during the dark period or by extending the duration of the light periods. Moreover, the critical day length varies depending on the species. For example, barley or wheat plants will only flower when the day length exceeds 12 hours, while spinach plants require a day length exceeding 13 hours to initiate flowering. Certain species like agrostis have an even longer critical photoperiods that requiring more than 16 hours of daylight to trigger the flowering. Here in this table, there are some example of the long day plants. It includes the various monocots such as the barley, bendgrass, oats, orchard, grass, ryegrass, timothy and wheatgrass, along with the certain dicots that includes the cabbage, clover, cornflower, dill, henbane, hibiscus, petunia, radish and spinach. These plants require longer periods of daylight, typically around 14 to 16 hours in a 24-hour cycle to induce flowering. Then moving ahead, here we have the slide related to that of the day neutral plants. These plants exhibit flowering across a wide range of photoperiods spanning from 5 hours to continuous exposure for 24 hours. Some of them have obligate requirements, meaning they strictly require a specific photoperiod for flowering, while others have facultative requirements indicating that they can flower under various photoperiod conditions. Examples of such plants include the tomato, cotton, sunflower, cucumber, peas and certain varieties of tobacco. Moreover, plants having absolute requirement for a particular photoperiod for flowering are called qualitative photoperiod types. This figure basically illustrates the regulation of flowering by photoperiods involves the transition from vegetative growth to the flowering, which is influenced by changes in the critical duration of the darkness. A reduction in the critical dark period prolongs the vegetative stage of the plant, delaying flowering. Additionally, exposure of pulse of light during the dark period effectively suppress the flowering. These mechanisms highlight the intricate control of flowering by photoperiodic cues, where alterations in darkness duration and light exposure play a crucial role in determining the plant's developmental stage. Some plants have evolved a strategy to avoid ambiguity in seasonal changes by distinguishing between shortening and lengthening days, known as dual day length plants. These plants can be classified into two categories. One is the long short day plants and another is the short long day plants. The long short day plants are inherently short day plants but require exposure to long day during their early growth stages to facilitate subsequent flowering. Example of long short day plants include the Bryophyllum, Kalanchoe and night blooming jasmine, which typically flower in late summer and fall when the days are becoming shorter. On the other hand, the short long day plants are typically long day plants but need exposure to short day during their early growth stage for optimal flowering later on. The examples of short long day plants include white clover, Canterbury bells, and Echeveria, which bloom in early spring in response to lengthening days. Moving ahead, now we will discuss the critical role of the dark period. Plants do not rely on measuring the relative length of day and night or the overall duration of the photoperiod to regulate flowering. Instead, they assess the length of the dark period as a pivotal factor in this process. This concept was demonstrated by Casey Hamner and J. Bonner in 1938 through experiments conducted with xanthium plants. In a 24-hour cycle comprising alternating periods of light and darkness, xanthium plants only initiate flowering when the duration of the dark period exceeds 8.5 hours. Conversely, when exposed to 16 hours of light followed by 8 hours of darkness, xanthium plants remain in vegetative state, underscoring the significance of darkness duration in flowering regulation, as you can see here in the figure. Similarly, the long day plants have a requirement for a dark period that is shorter than a specific critical maximum duration to induce the flowering. In these plants, if a short burst of light occurs during an otherwise non-inductive long dark period, it can reduce the necessary duration of the darkness to below the critical maximum, allowing flowering to take place. The capacity to gauge the length of the dark period is essential for plants practicing photoperiodic timekeeping, as it regulates the timing of significant developmental events like flowering. This figure shows the flowering response in long day plants and short day plants in 24-hour cycle, that is a photoinductive cycle. In nature, plants are subjected to photoperiodic cycle comprising alternating intervals of light and darkness throughout each day, as illustrated in this figure. These cycles play a critical role in regulating flowering in plants, is known as photoinductive cycle. Conversely, if a photoperiodic cycle fails to induce flowering in a plant, it is classified as a non-photoinductive cycle. These distinctions highlight the significance of light-dark cycles in determining the onset of flowering and demonstrate the plant's sensitivity to variations in photoperiodic conditions. A photoperiodic cycle consisting of 16-hour light and 8-hour dark period generally induce the flowering in long day plants, while a cycle consisting of 8-hour light and 16-hour dark period induces flowering in the short day plants. So, moreover, this figure illustrates the categorization of plants according to their response toward the length of day and night. So, moving forward to the photoperiodic regulation of flowering, as you can see here in the figure, the short day plants, also known as long night plants, bloom when the duration of darkness surpasses a critical threshold. The onset of flowering is inhibited when the dark period is interrupted by short exposure to light, known as the night break. Long day plant, also referred to as short night plants, bloom when the duration of darkness falls below a critical threshold. For a certain long plant, flowering can be induced by reducing the length of the night through a brief exposure to light, that is known as the night break. The number of photoperiodic cycles required to trigger flowering in plant varies depending on the species. For instance, species like Xanthium, Stromarium and Forbitus nil require just a single short day photoinductive cycle to initiate the flowering. In contrast, plants like Salvia occidentalis, classified as short day plants, may demand a minimum of 17 photoinductive cycles of flowering to occur. Similarly, Plantago lanceolata, categorized as a long day plants, exhibit a need of 25 photoinductive cycles to achieve optimal floral response. Interestingly, if a plant is restricted to a non-photoinductive cycle, only 15 cycles are required for flowering initiation. This phenomena suggests the accumulation of a specific factor responsible for the flowering response during the inductive cycle, thereby influencing the plant's flowering behavior. An important aspect underscoring the significance of the dark period is its susceptibility to disruption through the brief exposure of light, referred to as night break. Unlike interrupting a long day with a short dark period, which does not negate the effects of the prolonged day-night, a night break can render the dark period ineffective in stimulating flowering. Night break treatments of just a few minutes have been found to effectively prevent flowering in many short day plants, including Xanthium and Forbitus. Conversely, promoting flowering in long day plants often requires significantly longer exposure to night breaks. This observation highlights the differential sensitivity of plants to alterations in their photoperiodic environment and underscores the importance of understanding these responses in the context of plant physiology and development. The effectiveness of a night break in altering flowering responses varies significantly depending on the timing of its administration. Research indicates that irrespective of whether a plant is classified, exerts its greatest impact when administered close to the midpoint of a 16-hour dark period. As you can see clearly in this figure, the time at which a night break is given determines the flowering response. When given during a long dark period, the night break promotes flowering in long day plants and inhibits flowering in the short day plants. In both cases, the greatest effect on flowering occurs when the night break is given near the middle of the 16-hour dark period. The long day plant Fuchsia was given a 1-hour exposure to red light in a 16-hour dark period. Xanthium was exposed to red light for 1 minute in a 16-hour dark period. Moving to the next, the perception of the photoperiodic signal triggering floral induction primarily occurs within the leaf rather than the shoot apical meristem, as demonstrated by the Russian physiologist Chelyakhin in 1937. Chelyakhin experiments with Chrysanthemum morifolium, a short day plant, revealed that flowering occurred when only the leafy portion of the plant experienced short days, while the apical meristem and defoliated shoot remained under long day conditions. Conversely, when the conditions were reversed, with the upper defoliated portion exposed to short days and the leafy portion subjected to long days, the plant remained in a vegetative state. In another series of experiments involving short day plants like Perilla and Xanthium, it was observed that these plants could be prompted to flower even when all leaves except one were removed, and that single remaining leaf was kept under short day condition. Furthermore, when leaves taken from the induced plants were grafted onto non-induced plant, it led to the induction of flowering in a previously non-induced plants. This suggests that photoperiodic signal triggering flowering is primarily perceived and transmitted through the leaves, as evidenced by the ability of a single leaf kept in short day conditions to initiate flowering in otherwise leafless plants, and the transmission of this floral inducing signal through grafting from induced to non-induced plants. In additional experiments, leaves removed from Perilla frutescens, a short day plants, were exposed to a photoinductive cycle and then regrafted onto non-induced plants, resulting in the induction of flowering even under non-inductive long day condition. This effect persisted despite the continued exposure of the plants to long day conditions, demonstrating the resilience of the floral inducive signal. Chelyakhin's investigations also revealed that a leaf in its rapid growth phase is most receptive to perceiving the photoperiodic stimulus when it reaches approximately half of its final size. Moreover, in experiments involving interconnected xanthium plants through grafts, all plants underwent flowering, even if only the first plant was subjected to short days. From these findings, Chelyakhin proposed the existence of a floral stimulus hormone termed the Florigen, suggesting its potential to diffuse through graft union and mediate the transmission of the floral inducing signals. Grafting experiments were conducted between plants within the same botanical family but with differing photoperiodic requirements, exemplified by the short-day plants Nicotiana tabacum and the long-day plant Hyoscyamus niger. Notably, Hyoscyamus niger exhibited flowering under short-day condition when grafted to tobacco plants maintained under short days, while the grafted tobacco plant flowered under long-day condition when grafted to Hyoscyamus niger that kept under long days. These cross-grafting experiments suggested a potential universality of the floral stimulus across different photoperiodic classes. So, Chalahian posited that this floral stimulus likely functions as a hormone which he termed Florigen. He hypothesized that Florigen is synthesized in the leaves and subsequently transported to the shoot apex where it triggers the initiation of the flowering. So, this is all about the photoperiodism. Thank you.